making sure my phone is switched off so it doesn't bother us. Okay, so the whole idea behind today's lecture is going to be basically cell communication and how cells kind of not just communicate with their partners right next to each other, but with the larger tissue and then the larger organism that they are kind of hanging out in. So there are several ways that cells are kind of getting anchored where they are, and then several ways that they are communicating with each other and connecting to each other. And we're going to kind of talk about each level of those connections one by one. We're going to start off with kind of the most basic, the most, kind of spread out of these connections, connective tissues, and these are your extracellular matrix and the connective tissue. So this is kind of the scaffolding, if you can think about it that way. So it's, the scaffolding like you would see around a building, kind of providing the general shape and the structure for all the cells to accumulate in and get settled in and then kind of start working together in. We are then going to shift to talking about how individual cells are getting attached to each other and communicating at some level to each other, in addition to all those proteins that are there. And that's going to be by looking at epithelial sheets and cell junctions, various type of these connections that form between cells that are adjacent to each other. And then finally, we're going to go back and look at the process of cell differentiation again and kind of talk about stem cells and tissue renewal and how cell differentiation not just happens at the start, at the beginning, when things start to develop, but that it continues throughout your lifetime at various levels in various organ system to kind of continue the survival for that organ system and continue its functionality over time. So when we look at multicellular organisms, obviously majority of these cell communities and the cell-cell interactions, that's where they come into play, is in multicellular organisms. Um, our multicellular organisms are going to be typically designed so that they are divided up into organs, and then individually they would be organized into tissue systems. So if you look at just vertebrates, you have your nervous system, you have the muscular system, you have your epithelial layers, and then you have your connective tissues that are kind of serving as the backdrop or scaffolding for all these other tissues to hang out with. Now, each individual tissues, if we were to look at them more in detail, they are going to be made up of various types of cells coming together and then getting embedded into some kind of extracellular matrix. Our general body cell structure is designed in a way that cells typically require some type of attachment point before they will survive. They will be able to, settle down, grow, and survive. And so, unless it is one of our blood cells, which can be just, that can survive in without adhesion, most of our cells need to be adhered to some type of a scaffolding. The extracellular matrix is what provides them that glue or that scaffolding to attach to. Now, the extracellular matrix itself is secreted from cells, um, typically not the cells that are part of the function for that organ or that tissue, but rather specialized cells like fibroblasts, whose job is to secrete the materials needed in that extracellular matrix. Um, in addition, there are proteins and some substances secreted by other cells that are part of the tissue as well, that will be combined with that fibroblast and the extracellular matrix material secreted by the fibroblast in order to create a very strong kind of gluey structure or a concrete structure for the cells to work in. Now, in addition to the ECM, you also have cell adhesions, or kind of like cell-cell interactions that are additional kind of glue or additional contacts to keep these cells together and to keep them in the structure that they need to be. So that's kind of like, if you had two separate Legos that were individual cells, they kind of stick together in a particular way with the help of these cell junctions and cell adhesion molecules. So looking at a couple of examples from these, Here you can see how cells, different types of cells may be organized into tissue systems. So here you have a kidney section. So it's a section from one of the ductal tubes from there. And you can see that in that tissue from the kidney, when you cross section it, the tubes portion, you have these epithelial cells, ductal epithelial cells, organized in a very particular way to create those tubes. And then different types of cells around it are organized in a very different manner that make up the tissue around those ducts to maintain the structure of the kidney and to keep those tubes in the place they need to be. And that will be composed of multiple types of cells, including fibroblast and the extracellular matrix, making up that scaffolding. So some of that scaffolding is actually visible when you look at this clearly, right? All this kind of orangey stuff where there are no cells is extracellular matrix proteins that are just kind of setting there as a gluey structure. And then you see these individual cells with the nuclei inside. So you know that that's where an actual cell is sitting down. On this side, this is obviously just a drawing, but this again is showing you a lumen of a gut, again, this time a transverse section of it. And you can see going from top to bottom, there are many different types of layers within this lumen, each layer composed of a different type of tissue system. And then each tissue in there composed of various types of cells. They don't necessarily contain a single cell type within it. So you have this very organized epithelial layer on the top that is then underneath it, you'll have this really large connective tissue layer, which will contain a lot of extracellular matrix protein, providing the structure to that space, along with fibroblasts that are going to be secreting this material to help glue these cells together to keep them in their place. Underneath it, there are two separate layers of fibers um, associated with muscle structures, right? That will help this structure contract and relax to release the contents from the lumen into the gut or out of the gut. And so you have these smooth muscle cells, some that have very different shapes than others, right? You have these circular fibers versus the longitudinal fibers. So these are essentially muscle cells that are organized in different orientation within that space to create that final structure. And then you have additional connective tissue layer on the other side, along with the final epithelium boundary. So on both edges of this lumen, you will see epithelial layer, and in between will be your connective tissue along with the muscle to help it function the way it needs to do. So the first thing that we're going to be talking about is extracellular matrix, obviously, and this connective tissue that helps create the scaffolding and the cement to keep all the cells together and to keep them organized in their particular state. And we started by talking about plant cells because plants have really strong structural support in the form of their extracellular matrix and in their structural components. So the plant cell walls are a type of extracellular matrix, but all cell walls are not the same. They are secreted by the plant cells themselves, and depending upon what type of composition that cell wall is made of will dictate what type of structure that particular cell is making. So you can have a thick and hard cell wall, and that would be what our wood and stems are composed of, right? And then you can have thin and flexible cell wall, a little bit more malleable, softer cell wall, which would be what you would find in the cells of the leaf, for example, or in some of those thin tendrils that are part of the plant structure. So over here, you're seeing a cross section from a plant stem where you can see both types of cell walls being made. The wood, the thick and hard ones, contain a lot of cellulose, which is a type of cell wall, extracellular matrix material. And so those are stained in the blue dye over here, the blue fluorescent dye over here. The softer, more malleable and flexible cell wall is made up of pectin. So that's what you're seeing on the top portion of this particular cross section. is that this section on the top, the beginning portion of that structure has more malleable, more soft components, and that's your pectin. And underneath is more stronger component, which is your woody cellulose, more stronger, thick and hard cellulose. Now, typically, the new shoots that come out will be higher concentration of pectin and less concentration of cellulose. And as they become, as they grow into their final form, their growth slows down and they become kind of stationary as far as growth is concerned. They are, like equivalent of senescent cells. Then they start to deposit more and more of the cellulose to make it more hard and rigid. Um, so typically cells that are still growing and developing and replicating, dividing, actual active growing shoots will be higher concentration of pectin. Now in this particular image, you are only staining for those pectin and cellulose, so you're not seeing any of the cells. It just shows up as holes. But on the this electron micrograph image, you can see both. So here you can see, and this is from a root. So it is from that growing portion, a root tip of the plant. So from the same plant actually. But here, remember that in that root, again, if you're going towards the tip, that's the edge that is actively moving, actively growing. So you'll have less of the cellulose and more of the pectin. But you can clearly see in this case the cell walls on the outside of the cell structures and then the cells inside, right? And you can sometimes even see the clear vacuoles in there and some other components that are part of that cell. Cool? Questions? Okay. Okay, so now let's look at why cellulose is so hard and woody, and why is it that it provides the cells more structure and more rigidity. So the cellulose is actually, at the end of the day, it's just carbohydrates that are joined together in these long chains, right? So cellulose molecules are basically made up of, it's a polysaccharide chain at the end of the day, where you have a beta 1,4 linkage between adjacent units of the sugar molecules, and they're just made up into long chains. Now, 16 of these molecules together make one chain of the cellulose fiber, and they are then lined up together. 16 different ones of these, this long chain of cellulose molecules, 16 of these are bound together through hydrogen bonding to create a structure that we call a cellulose microfibril. So this is very similar to, if you go back and think about some of these other cytoskeletal structures that we've looked at, right? In the form of, for example, the intermediate filaments and microtubules and actin fibers. So it's similar to that in the way it is organizing, that many of them are coming together to create a stronger fiber or a stronger structure. Now, these aren't wound around each other. These are not actual covalent linkages, but rather just hydrogen bonding that are keeping the various chains of cellulose molecule together in this microfibril structure. Now, these microfibril structures are then interwoven in your in the wall of the cells. And so if you were to look at a plant cell, down here is going to be the cytosol and the rest of the cell, the plant cell itself. Outside the plasma membrane is where these are going to be kind of lining up. So think about that, laminar structure inside on the plasma membrane, right? You had structures made on the inside of the plasma membrane with the actin and the spectrin molecules combining together. This is kind of the same kind of scaffolding, but on the outside. And so this is what you have, what you call a primary cell wall. It is made up of a bunch of these microfibrils, cellulose microfibrils that are kind of interwoven into each other, kind of like when you create some type of a basket structure, a weaving structure. And then they are joined together with what we call cross-linking polysaccharides, other types of sugar molecules joined in long chains that are connecting these individual microfibrils to each other by their interaction. And then remaining space that is left behind is kind of woven in with pectin, which are the more thin, flexible molecules, so they can kind of weave in and out of different structures. So combined, it gives you a lot of strength in that structure, so you can put a lot of compression without causing damage to that cell, right? Yes, no, maybe?
Yes.
Yes, and how about like the new shoot? It will have more pectin at this situation?
Yeah, so when you have a new shoot, you'll have less of these microfibrils and more of the pectins, which is why they're a little bit more flexible, but then easily, they cannot take as much of that tension, right? As much of the kind of pressure, compression as the harder, older shoots can.
Okay.
So now the orientation of these microfibrils, remember we talked about how they're kind of, stacked and they're in various orientation, is actually what's going to control which way that shoot is going to grow. And so the plants, cells actually control how they lay down the cell wall, in what orientation the microfibrils are going to lay down into that first layer of structure. And with that, it can control its shape and direction of growth. So we're going to talk actually in the next slide about how they control it exactly. But right now, what you need to know is that, these structures and these microfibers can lay down in a way that the plant grows up, right? Grows in a straight upward direction and grows taller, or they can lay down sideways so that the new shoots grow outward from the plant and the width of the plant is growing in that way, right? Regardless of how they are laid, the one thing that is constant is that the turgor pressure on that plant is going to be uniform in all directions. So it will have still uniformity in the type of compression it can take, regardless of its orientation, all over the shoot. And so the way the plant dictates how they're going to control the direction of this growth is by anchoring these new cellulose microfibrils inside the cell to microtubules. So those are your main structural components inside, maintaining the shape, right, inside the cell. And so you have these special microtubules right on the inside of the plasma membrane that are anchored into the plasma membrane through integral proteins. And here you can see an example of that. So these microtubules that are going to be the attachment points for these cellulose microfibrils are going to have attached these integral proteins all along them to control where they stay, so they don't have a lot of movement in them. And then they are going to be in turn also connected to other proteins that act as connector proteins to and a little kind of a transporter to an enzyme that is integrated into the plasma membrane that creates cellulose. So this little enzyme is called cellulose synthase. It's a complex of a bunch of different proteins coming together in this structure to create almost like a pore type conformation. It's connected intracellularly to a connector protein that in turn is anchored to the microtubule. The whole job of this particular enzyme is to create cellulose molecules or cellulose microfibrils. It does so by taking glucose from the cytoplasm. So it is, importing glucose into it. It's using it to join them together into those long cellulose chains that can then self-assemble into those microfibrils that then will come out of those pores, and they will obviously then be part of the existing structure that's already there. Now, the orientation of these microtubules is going to dictate which way that tube is going to go, right? So you can think about it this way, that if the microtubules are oriented in the way that all the microfibrils grow out towards the right, then the shoot is going to go out towards the right. If it is oriented so the shoots are going up, then the shoot will, the plant will elongate and become longer and longer. So that is how the cell controls which direction these shoots are going to grow by controlling in which direction these cellulose microfibrils are being deposited in that cell wall. Taylor looks confused.
No, I'm not. It's just really bright from squinting.
Ah, gotcha. So, basically the microtubules are the orientation of the microtubules inside the cell is what's going to dictate the outshoot that comes out in the form of the cellulose molecules. And those cellulose microfibrils, the direction that they move in will then dictate the way the plant grows. In animal cells, the connective tissue is going to work a little bit differently, right? So in plants, it's more structured, it's more organized, and it's because that's what they need to maintain their structure in the space. In animal cells, the connective tissue is usually produced in a really large amount, and it is usually kind of like this gummy, sticky glue substance that is present in the scaffolding of that organ system to maintain the cell's space where the cells are going to be in. The ECM can take a lot of mechanical load because of the way it is organized and structured, and we'll talk about what it is that allows it to carry such mechanical load. And then the cells that are embedded within it can join to each other as well as join to the ECM itself in order to maintain their structure in the space. Inside this ECM, again, you have something very similar to what we saw in plants. So in plants, you had these cellulose microfibrils. The animal cells instead have collagen fibrils that are organized into bundles similar to cellulose polysaccharide chains. And again, their function is going to be to provide tensile strength to the connective tissue so that they can take certain amount of compression without getting damaged. In this case, they are formed a little bit differently. A single collagen polypeptide chain is just going to be by itself, but typically a single collagen molecule, quaternary structure of this molecule is formed by 3 polypeptide chains coming together to form a coiled structure through their bonding interactions with each other. And this structure already you can see is going to be a little bit stronger, right? Because there are 3 of them twisted against each other, kind of creating a little bit of a rope-like structure that you've seen before in intermediate filaments as well. And then these triple-stranded collagen molecules, when activated or when cleaved in just the right way, can self-assemble to form these larger fibril structures which contain multiple fibers all bound together in a very tight-knit structure. And so a single collagen fiber will have a lot more of these connective tissue collagen fibrils joined together to create a very strong rope-like structure. Now, this is kind of like, if you had a single pencil together or 3 pencils together versus a whole bunch of pencils together. So it's going to be a lot harder to break it, a lot harder to destroy it. Now, if you see a cross-section electron micrograph of tissue, connective tissue, you will see that, again, just like we saw in the plants, these collagen fibrils are oriented in both directions to create this weave-like structure to give it extra tensile strength. They're not all oriented the same way. Some are going longitudinally and some are transverse. So you can see right here, these longitudinal ones, right, all along, and then you see some where you can see the ends of those collagen fibers, and you can see individual collagen molecules inside, which are the triple-stranded coiled collagen molecule. In there, multiple ones of them in a single bundle. And you can see that there is some variability in the number of these molecules that are within a bundle. They're not all going to be made up of 100 molecules together or 50 molecules together. Some are smaller bundles than others, and that's dependent on the space that they are in and what type of other interactions that they need to make in that space to create that organ system. And then also within it, you will see your tissues that are then organized within that space. So the collagen fibrils come from the collagen polypeptide. So a single collagen polypeptide contains that central portion, which is going to be part of the collagen fibrils, and then it has tails on both the amino as well as the the carboxy terminal side that kind of stick out, that cannot make any interactions with each other. And so that structure in its full length is what we call a procollagen precursor. This is what is secreted by the cells in vesicles once it is made. So these are your essentially individual triple-stranded collagen molecules without the cleaved ends. So they are still not able to join, self-assemble and join together. So that this is done so the collagen fibers don't self-assemble inside the cell, and then the cell will die essentially because it won't be able to do anything. Instead, they remain this way until they are secreted out into the extracellular space where there are specific proteinases, right? Specific proteases that will cleave these terminal ends on both sides, leading to the final collagen molecule that can self-assemble into fibrils. So it cannot self-assemble into these larger fibril structure until and unless these ends are a protease cleaves those ends and releases them from that space. So the mature collagen, once it is created, is what's going to self-assemble into the fibril. Now, depending on the space it has, it can make a fibril as little, as narrow as 10 nanometers wide and as wide as 300 nanometers wide. So they can have a range of different strengths depending on the size of these fibrils at the end of the day. Now, cells will organize this collagen that is secreted, sometimes in a little bit haphazard way, but many times in a very oriented fashion, especially in special organ systems like your skin. You will see them organized in a very specific way, um, organized way to maintain the shape and the uh, type of skin structure that you need. So it's again, the most of the collagen that we have is going to be secreted by fibroblasts, and those fibroblasts, you can see them inside the extracellular matrix as well. They're kind of, stuck in that space. Um, it's deposited in a very organized manner. So you will have bands of fibers going one direction and then bands going the other direction. So you can imagine if this cross section was cut the other way, it will give you that kind of basket weave like fashion, right? It will have those little squares in there because of the way that they are organized throughout that structural space. Um, so the fibroblasts are able to move within that space in that sticky goo. Other cells are not. Other cells are adhered more strongly to that structure and are less mobile in the space when they are arranged there. So skin, like I said, especially are arranged in this woven pattern. So if we were to cut them in one cross section, you will see bands on one side and a kind of basket weave pattern on the other by alternating orientation of the layers. And this is to create, again, a very strong structure as our boundary so that it is impermeable to outside kind of toxins and outside agents that would be harmful to us. And also it gives us extra strength to that space so that our internal organs are then getting protected by them. In tendons, on the other hand, you will notice that this will be arranged differently, where all of them are going to be aligned in parallel bundles instead of in this woven pattern like you see in the skin. Now, cells, when you look at actual cells that are not secreting the extracellular matrix, they hold on to the matrix with the help of other proteins that are on their cell surface, in the plasma membrane, and additional proteins that are part of the ECM in order to create a strong binding to the structure and maintain their space in that organ. So one of the proteins that they use is fibronectin. Fibronectin are these kind of, hook-like proteins. They're long protein chains that have attachment sites both to integrants that are on the cell surface of the cells and extracellular binding sites to the collagen fibril themselves to anchor the cell to the collagen fiber. They in turn will bind, so these are extracellular proteins at the end of the cell a day. They bind to integrins that are on the plasma membrane of the cells that are trying to attach to the ECM, and then in turn also bind to the collagen fiber. So if you look at this, this is a multi-protein complex, right? So you have your collagen fibers that are binding to the fibronectin ends, the end terminal of that. And then you have the C terminal or some part of the fibronectin binding to the integrins that are bound to the plasma membranes. Now, sometimes that's all the interaction they have. Other times we will notice that there are further anchor points within the cell to even more strongly attach the cell into the space and make it even less mobile by creating an anchor point with adapter proteins from the integrins cytosolic site to an actin filament that is part of its structure inside. And so now it is, you can imagine a very strong interaction that will be pretty much immobile. So the cell is stuck where it is. It's not going to move much, right? If you didn't have this internal, binding site, then the cell is going to be a little bit more mobile. It's going to move around a little bit more. It's going to shift as the collagen fibers shift or the cells around it shifts. So depending on the cell type, you will notice that sometimes you have a lot of anchorage points to maintain that structure in a very strong way so that the cells are not moving in that space at all. Now the integrins can switch to, the integrins usually work as dimers. So you have usually an alpha and a beta subunit, and they can switch to either an active or an inactive form depending on what type of environmental stimulus they receive, so that they can either bind to the extracellular matrix or they may not be bound to the extracellular matrix. So in a normal case, if there is a situation where they need less binding to ECM, you will not have the active form of the integrins, and they will be bound to each other rather than to the ECM. However, when there is a need to create a strong anchor point, you will have these cytoskeletal adapter proteins binding to the beta subunit, activating it, which in turn will activate the integrins themselves to be in a more stretched out condition where they can now bind to fibronectin and then bind to collagen to maintain that strong bond and create that anchored cell structure that you were looking for. So extracellular matrix and connective tissues basically are working very similar to how we saw cellulose in plants. They are going to be arranged in different orientations to provide tensile strength to the cell. You are also going to have other proteins like fibronectin and other types of polysaccharides to fill up the extra space inside that ACM to maintain a strong structure, a kind of like, concrete-like structure that will resist compression, and also provide anchorage points to cells to bind to. And, proteins that will bind both to ECM as well as the cells to maintain that strong structure. Questions?
I had a question about the integrins, like the
Yes.
slide before. Are they always like either connected strongly or not connected, or their situation, and what are those situations when they need to connect strongly to the core?
So it depends on the organ system. Some organ systems, the cells are always going to be anchored to the ECM and they're not going to be coming off. And so this binding will always be strong. However, imagine a scenario when a cell is about to undergo cell division. At that time, it needs the flexibility of its plasma membrane in order to do appropriate cell division and cytokinesis. At that time, actually, the cells lift off the ECM. They are no longer attached for the time that they are undergoing mitosis. And so at that time, everything is going to be like this. All the binding sites are going to be closed up. The cytoskeletal structure that normally maintains those binding will not be present. They will all be instead involved in the actual mitotic spindle and the mitosis that's happening. And the cell will be lifted up like a ball, will divide. And then once it has divided, it will go back and attach back to the ECM. So that's a very classic example of important example of how integrins switch to an active versus an active form in order for the cell to perform the function it needs to do, and then revert back to binding to the extracellular matrix when it's needed. What happened? 1 second. Okay, come here. 1 second, guys.
You're fine.
Okay. So did that make sense, Osana? Did that clear
Yes.
up it a little bit?
Yes, it does. Thank you.
You're welcome. Okay, so let's look at some of those other things that are present inside the cells, right? Inside this extracellular matrix to make it the super gooey area that other cells can attach to. So one of the things that are going to be present there is what we call glycosaminoglycans. So these are essentially large sugar molecules, and these are usually charged sugar molecules, negatively charged sugar molecules, that are going to in turn associate with proteins in this ECM, in the extracellular matrix, and they can also associate with other sugar molecules that are attached to proteins to form this really complex, super large macromolecule. Now, because they have all these negative binding sites, they provide attachment points to cell surface protein receptors with other sugar molecules attached to them to bind to them. And it, again, creates a strong binding that will then maintain the structure and placement of those cells. These proteoglycans, if you were to look at them inside the cells, you can see how you have these really large chains of glycosaminoglycans that have then aggregated other types of sugar and linker proteins to them, giving this, kind of centipede-like look, right? So you have the main sugar molecule, you have other proteins with sugars attached to them, attached to it. They're all linked to it with linker proteins. And then this complex structure just provides a lot of sticky places for other proteins to attach to them, right? So one of the molecules that you'll see in this space is hyaluronic. I didn't pronounce it correctly, but it is one of the main, and chondroitin sulfate. Those are two main components of this structure. Now, in our skin, in our hair, in our nails, especially in our nails and hair, you will see a lot of keratin sulfate and keratin aggregates as well that are also going to be part of this structure to maintain, again, further structure and provide further structure and stability. So these give great strength to the extracellular matrix and provide more resistance to compression from outside sources. And typically the harder, again, the structure, the more of these you will tend to find in those spaces. So hair and nails will have a lot more of these than some, let's say, skin, for example. So plant cells have tough external walls that are made up of cellulose microfibrils. The more flexible ones in there are going to be formed from pectin or a combination of cellulose and pectins. And these are going to be what provides the plant cells their tensile strength. In animal cells, this connective tissue and the same tensile strength comes from a combination of collagen and these other glucosaminoglycans and the polysaccharides that are secreted into that extracellular matrix to provide that tensile strength. The fibroblasts are the cells that are organized to secrete collagen specifically to create those collagen fibrils. And integrins in the protein, integrin proteins in the plasma membrane of the cells is what couples with other proteins into the collagen fibrils to create the structure where they can bind and work together to in that space. So the next part that we're going to talk about is epithelial sheets and cell junctions. So now we're going to talk more about the cells that are inside the extracellular matrix, inside the scaffolding created by that, and look at how are different ways that they can be organized together, and then how are they going to be interacting with each other and with the environment. So we're going to start off with that outside layer of tissues and cells and organs, which is usually your epithelial layer. And that epithelial layer is extremely important for the integrity of not only that tissue system and the organ that they are part of, but also they are extremely important in maintaining the structure and function of that cell, of that organ system, because they are essentially who is that, they act, if you think about it, they are like the plasma membrane of a cell, right? That's the same idea. They are the ones, they are the frontline workers that are receiving the signals from the environment, from the surrounding tissues, from the surrounding cells, and transporting them inside so that the cell can, that organ system can function the way it needs to. Now, there are four different ways that epithelial sheets can be formed based on the organ they are in and the function they need to perform. All of them are going to be immediately attached to a laminal sheet of, which is another way, part of extracellular matrix, a laminal secured, they are secured to that basal lamina, which is in turn then connected to the main connective tissue. So one way that you will see these epithelial cells joined together are in columns. This is especially important in like our lumen gut, in our intestinal lining, in our lungs, anywhere where you have different types of information getting input from one side and then sent out to the other. So you will have these cells in columns where the basal side of the cell is going to be very different than the luminal side, than the one on the top, right? The one that is actually interacting with the environment is going to be very different than the one that's interacting with the connective tissue and the cells further inside that organ or tissue layer. Another way is squamous orientation, where the cells are just kind of making a boundary or a lining of your structure that they are in. And then they are immediate, it's just usually a single layer of cells. And then immediately afterwards, you have other layers of very different types of cells that are part of that organ. This is something that you may see in your blood vessels and your lymphatic vessels and those type of structures, that where you'll have a single layer boundary and then you'll have other types of cells inside. Then you may have cuboidal cells. Again, these are also cells where the way they're organized are more tight-knit community. They're going to be more strongly adhered to each other than squamous cells. So both columnar and cuboidal are similar in some ways. They have very strong attachments to each other. They have a lot of junctions to interact with each other. And they both have kind of a top side and a bottom side, where the basal side of the cell is going to be a little bit different than the luminal side. So they will be having some different, if you looked at the plasma membrane, the protein composition in the luminal side is going to be very different. The kind of receptors that are present on the top side are going to be very different than the ones that are present on the basal side of that same cell. And that's for them in order, like glucose transporters we've talked about and other such things, where you have very different environments to look at on the top versus the bottom side of the cell. And they have to react to very different types of stimuli. And then finally, you have what we call stratified cells, where it's large organ systems and large layers that are dependent upon these epithelial layers. So our skin, for example, they'll be all kind of spread out all along it in large sheets-like manner. So epithelial sheets are polarized, like I said. In many cases, they have an apical and a basal side, or luminal and a basal side, depending on what type of cell you're looking at. And they are going to be, they will have very different environment on the top side versus the bottom. So they will have different cell surface receptors to interact with those two sides of their environment. They are usually attached with strong junctions. There was something. So they will have very strong attachment points, usually at the apical side, anchoring them together in a very strong concrete manner, so that they can communicate very easily with each other on that side. And then on the basal laminal side, they may have a little bit less connections and less interaction, less junctions to keep them together, but they will be interacting with the connective tissue on that side. So um, the basal lamina basically is what's supporting this layer of epithelial sheets. It's providing the anchor point. It's like its own little uh, extracellular matrix, a simplified matrix connection before the underlying major connective tissue is starting. And that basal lamina is a place for them to kind of get anchored to before they interact with the other cells. So here, for example, this thin layer of, layer in between this kind of disorganized looking connective tissue and these epithelial layer cells, these tall epithelial cells, is the basal lamina. It's a very thin layer of connective tissue of specific glycosaminoglycans and sugary proteins and connective tissue like material, ECM like material that's connecting, providing an anchor point for these cells to get attached to. Now, our gut lumen is an example of where these highly polarized cells are needed for transport of nutrients and glucose molecules, right? They're interacting with this super high concentration of glucose molecules, they're taking them in, and then they're transporting them on the basal side to the other so that it can be taken to the rest of the body, or in the intestinal lumen rather. That's what you would see. So these are examples of those. They may be inside that epithelial cell or sheet or layer. You may have other types of cells occasionally. Like in this case, you may have some of these cells specialized to secrete mucus or to have other type of specific functions, maybe secrete digestive enzymes or other things that can then be secreted into the internal or outer surface. So here you have these goblet cells that are integrated into this epithelial layer, that are your mucus cells that are going to be secreting the mucus that will come into your gut and kind of help the digestive process and protect the cells from the acids that are secreted. On the apical side of this surface, you will usually see very different biology or structural phenotypes. Many times they will have these microvilli or other pseudopods or areas that can take in certain types of nutrients or certain types of molecules that can then be absorbed into the cells and taken by the body the rest of the way. So the way they are joined at that apical side, right, on the top side of the cell, away from the basal side, are through tight junctions. So this is a specific type of interaction between two adjacent cells that are termed tight junctions, which causes some of the cytoplasm to be shared between all of these cells. Because if you look at the junction on from its side, you can see that there are spaces in this junction to share cytoplasmic space, right? So it will have the cytoplasm kind of going through in and out of that space to kind of share that space. However, it has little sealing points all along that junction to kind of hold those 2 points together. And this is done with the help of sealing proteins called occludin and claudin that join together to form this little dimer that can then seal on the other side with the adjacent cell and form a very tight junction. So it's like a double-sided tape that you put at a few inches apart, and then kind of stick them together. You'll still have a little bit of space that is not joined, but then you'll have these anchor points to hold them together. So if you were to look at a cell, a tight junction under an electron micrograph or under the microscope, you will be able to see those spaces where it is open and the spaces where they're sealed tightly against each other. Okay, beta. 1 second. Let me share him back with his grandma. Sorry about that. 1 second. Okay, so again, in this type of epithelial sheet structure, the only part where they're joining is that tight junction, right? And the cells may be joined with further tight junctions. In some cases, in cuboidal, you'll see a few more, but the other places, they're going to be completely separate from each other and then embedded into that basal lamina. So the tight junctions basically allow a place for these cells where they can share some cytoplasmic space, but it also serves as barriers to solute diffusion away from that cell, from that space, so that it's not moving molecules away to the basal side unless it is needed. In addition to that, there are several other types of junctions that link two epithelial cells together. Two most common ones are where they are attached to the cytoskeletal structure. Again, remember those actin filaments or microtubules are also serving as anchor points. Intermediate filaments are serving as anchor points as well. So there are two different types of junctions that require these cytoskeletal structures to bind to. One of them is adherent junctions, and the other one is a desmosome. So in adherent junctions, there are these special linker proteins called cadherin proteins that are embedded into the plasma. They are plasma protein receptors, cadherin proteins, that then in turn bind to linker proteins on your cytoskeletal structure in the cytoplasmic side of the cell. The cadherin proteins on the cell surface receptors, they can bind to each other on the, in the extracellular space to form a junction between two adjacent cells. So here, the plasma membranes are not right next to each other. They're not able to share information with each other except through interactions with receptors. So they're not linked the same way as tight junctions did, right? So these are tethered to the actin filaments, joined to the actin filaments inside the cell. Desmosomes also use cadherin junctions outside the cells, but the inside the cells, instead of actin molecules, they're going to be bound to intermediate filaments instead. So adherents and desmosomes both use cadherins as the cell surface receptor that binds to each other in the extracellular space between two adjacent cells. And inside the cells, with the help of linker proteins, will bind to cytoskeletal structure. However, adherents use actin and desmosomes use intermediate filaments inside the cell. Cool. So we'll talk a little bit more about adherent junctions, and I think then we'll be getting to the end of class time. So we'll then pick it back up from there for the next class. So adherent junctions are used by the cell to create a boundary between the apical and the basal side of the cell to create that polarity. So adherent junctions, and you can see some of them forming right here. So you have a tight junction where the cytoplasmic space can kind of talk to each other and interact with each other through these tight junctions. Then you have your cadherin molecules that are bound to each other at the cell with the adjacent cells, and they're bound inside in the cytoplasmic space with bundles of actin filaments all along. And by creating that space, this is separating that top basal side, the top apical side of your cell from the basal side, right? So this particular portion is usually the actin filaments are also going to be extending into the microvilli or any other extensions that the cell has. And it's going to also interact with that bundle of actin fibers joining those cells together. But these bundles allow the cells to then be tethered to each other and organized, and also help in creating that polarity within the cell. Okay, so we will stop here, and next time when we will start by talking about epithelial sheets and how we can then, once they are created in this very nice clean manner where they are have their polarity, they can then be used to create all kinds of interesting structures like tubes and vesicles to create the different organs and different systems that we need inside our body to do the work they need to do. Okay? Questions?
Okay, sounds good. No
Sounds
questions.
good.
No questions for me.
No questions. Okay. Okay then, guys. I will see you next week, or if I talk to you tomorrow. Okay?
Thank you, ma'am.
You're welcome.
Have a safe trip.
Take care. Thank you for your understanding. Baby will have his nanny back next week. He'll be happier. I'll be happier. Take care. You too. Have a great weekend.